All right, I'm Chris. I'm going to be talking about the effect of gut microbes on the development and severity of rheumatoid arthritis. Specifically, I'm looking at how it alters the pathogenicity of autoreactive antibodies and how the gut microbiome can be used to predict and prevent disease. So here's an overview of what we're going to be talking about. I'm going to start with an introduction to autoimmunity and rheumatoid arthritis, and then I'm going to transition into the microbial impacts of pathogenesis of rheumatoid arthritis. So rheumatoid arthritis is an autoimmune disease, and so autoimmunity is essentially a self-reactive immune process, which brings up this idea of self versus non-self. So we have cellular markers that identify our cells as being a part of self, and so that our immune system doesn't mount an immune response against those cells. Foreign invaders are marked as non-self. Essentially, they don't have those same cellular markers, and so we can mount an immune response to them. So autoimmunity can happen when you have the formation of autoantibodies, which are self-reactive, and that can happen at a lot of different points when you're generating antibodies, which is shown in this kind of flow chart. And so specifically in rheumatoid arthritis, typically it's when they fail peripheral tolerance, which is where you are forming antibodies that have failed peripheral tolerance is they're going to target tissue that's out in the periphery. So not in the cells that are producing these antibody creating cells. And so there are more than 80 known types of autoimmune diseases, and that number is increasing in terms of the amount that are having some sort of autoimmune component. And the estimates of prevalence vary widely while the number is increasing for affected people. So some epidemiology, it's a very widespread globally, and it's estimated to affect up to 1% of the population. So this image shows the global estimated disease-adjusted year life, which is essentially a measurement of disease burden calculated by the years of living with a disability and the years of life lost due to premature death. So the reason why there's a lot of this yellow in Northern Africa and Bangladesh and those regions is usually due to a lack of data coming from those countries. It's not necessarily that there isn't a high prevalence there. So first I want to real quick mention osteoarthritis. Osteoarthritis is another form of arthritis that is not autoimmune. It's just due to normal wear and tear. So I'm going to be talking about autoimmune rheumatoid arthritis. So it presents with chronic inflammation of the joints, warm, tender, and swollen joints, weight loss, morning stiffness that lasts longer than an hour, and rheumatoid nodules, which are essentially these firm tissue nodules that can happen at places like the fingers, wrists, and elbows mostly. So in terms of diagnosis, usually it's synoviitis or inflammation of the lining of joints for greater than 6 weeks. The size and number of affected joints are looked at. So rheumatoid arthritis usually impacts smaller joints first, and it's usually symmetrical. C-reactive protein, this is an opsonin, which is a protein that enhances phagocytosis. And this is a blood test marker that is usually used as a form of detecting if there's active inflammation going on. And then these last two, anti-citrullinated protein antibody and rheumatoid factor, are both self-reactive antibodies that are seen in rheumatoid arthritis and can be tested for in a blood test. But a quick note is that in juvenile idiopathic arthritis, which is actually a set of 12 diseases that are diagnosed before the age of 16, rheumatoid factor is only seen in about 2% of cases. So we're going to be focusing on adult rheumatoid arthritis for this presentation. As for other diseases that rheumatoid arthritis can then increase your risk for, cardiovascular disease, lung disease, lymphoma, and carpal tunnel syndrome. And then it also has an increased likelihood of developing a second autoimmune disease. So citrullination is a post-translational modification that's catalyzed by this enzyme known as PAD, or peptidylarginine deaminase. And some microbes can also citrullinate proteins like P. gingivalis, which is involved in periodontitis. And these microbes that have made these citrulinated proteins result in mimicking human citrulinated proteins, which is where we can end up developing anti-citrullonated protein antibody against self. And so we see that in about 60 to 80% of rheumatoid arthritis patients that they express anti-citrullonated protein antibody, and a similar percentage express rheumatoid factor. And so I mentioned here real quick collagen-induced arthritis. This is essentially the model of arthritis that's used in mouse studies. So that I'll mention quite a few times because a lot of the studies that I discussed today are on mice. So here's one of those types of pathways that have been proposed in terms of how rheumatoid arthritis can cause the formation of the autoantibodies and the symptoms that you see. So this Th0 cell, the naive T cell that's activated by something like a citrullinated protein, and that releases cytokines to activate other helper T cells to release cytokines and eventually reach B cells to proliferate and differentiate into plasma cells, which essentially plasma cells are they are antibody factories, and so these are going to be plasma cells creating antibodies that are self-reactive. But then that naive T cell also releases cytokines to activate CD4 T cells to release interferon gamma, which macrophages then get activated to release pro-inflammatory cytokines like TNF alpha, interleukin 1, and interleukin 6, which those activate osteoclasts, fibroblasts, and chondrocytes to cause the significant symptoms that we see, such as bone degradation and cartilage damage, and also fibroblasts build up the synovial tissue, which is why we see that swelling of synovial tissue as one of the disease symptoms. So currently there are three methods or groupings of methods for treating rheumatoid arthritis. There's NSAIDs and corticosteroids like ibuprofen and cortisone, which are used to to manage and treat whenever you go into a flare-up, which is a short period where disease tends to become active again. Disease-modifying antirheumatics like methotrexate, and biologics like there are three main categories: TNF alpha, IL-1, and IL-6 inhibitors that are all targeting those pro-inflammatory cytokines that were released to manage symptoms. And so I put a little asterisk next to the ones that I've taken for managing my rheumatoid arthritis. Currently, I'm taking adalimumab, which is also known as Humira. So now we're going to get into talking about the microbial impacts. So first off, microbial dysbiosis is an imbalance of the microbiome, or the collection of microbes that inhabit the human body. Typically, this stems from a loss of beneficial microbes, and it often results in disease. So some beneficial microbes might produce antimicrobials that protect the host from pathogens, while some will help with the intestinal barrier. So in this image, you can see there's a nice, on the left, which is gut homeostasis, you can see there's a nice thick layer of commensal bacteria that are there helping keep this functional barrier very healthy, and you have a normal immune response. When you have dysbiosis, you have this loss of beneficial microbes that results in a barrier defect, and therefore you can have a dysregulated immune response. And that's kind of where they're thinking a rheumatoid arthritis pathogenesis could stem from. So prior to visible arthritis, microbial dysbiosis develops, and it continues to develop with disease progression. So this kind of establishes this preclinical phase of disease where we could probably prevent significant disease through treatment. And so they found that depletion of the gut microbiome prior to this clinical phase resulted in about a 40% depletion of disease severity, reduced levels of serum inflammatory cytokines, and reduced levels of anti-type 2 collagen antibody, which is the autoreactive antibody during that mouse model that I mentioned earlier. But if you actually if you did this depletion of the gut microbiome during late phase CIA, you actually found an interesting finding, which is about a 50% decrease in disease severity, but there wasn't that much of a reduction in the actual levels of anti-type 2 collagen antibody. this is due to the fact that it only actually affected the function of the antibodies. It resulted in a significant reduction in their ability to activate complement system and produce inflammation. And so this brings us towards this important protein with regulating the intestinal tight junction, zonulin. And so zonulin can be stimulated to be released by intestinal cells due to your diet or due to microbes. And so what they found is when you have increased serum levels of zonulin, you have an increase in intestinal permeability and dysbiosis and inflammation. And so they looked at restoration of this intestinal barrier. And what they found is that if you restore it during the preclinical phase of arthritis, it inhibited the development of disease. So how exactly can we establish that an organism is causative of a disease? So this guy named Robert Koch performed experiments and designed a criteria to establish a causal relationship between a microbe and disease. And they became known as Koch's postulate. Basically, if you have a diseased host, then you should be able to take the organisms out of that diseased host, isolate and purify those. And then if you inject a healthy mouse or healthy host with that organism, it should cause disease, and then you should be able to isolate it again. That's essentially what they did in order to study this strain, Subdilogranulum. Isolate 7 is what I'll refer to it from now on. And it was isolated from the stool of patients at risk or diagnosed with rheumatoid arthritis and not found in the stool of healthy patients. So essentially what they did was they took the microbes from these stool samples and isolated them with anti-rheumatoid arthritis antibodies, and they found this isolate 7. Then they took that isolate 7 and injected it into a germ-free mouse, and from there they saw that it had spontaneous joint swelling, an expansion of Th17 cells, which increase inflammatory cytokines, and the production of autoantibodies that mimicked early rheumatoid arthritis. And so bacterial metabolic pathways that impact rheumatoid arthritis. So we're going to talk mainly about tryptophan metabolism. So tryptophan, it's an amino acid that's essential, so it has to be taken in through the diet. And so there are 3 different metabolic pathways that involve tryptophan: indole, serotonin, and carinine. Not sure if that's how you pronounce it, but we're going with it. So we're going to mainly focus on indole because that is the only pathway of the 3 that is relevant for the microbes that are in the gut. And so it's catalyzed by this tryptophanase A or tryptophanase enzyme, and we'll talk a little bit more about that in a couple slides too. So of the 3 pathways, indole metabolites were the most significantly increased in CIA mice. And so it had a really large increase, 528-fold, compared to those that were treated that had CIA. And so it suggests the shift of the metabolome that is associated with collagen-induced arthritis. And so they saw this in multiple models of inflammatory arthritis, such as that isolate 7 that I mentioned earlier. And so they also found that indole levels significantly correlated with the disease severity. So some microbial sources of indole in CIAs, there's firmicutes, so like lactobacillus, hydrogen anaerobacterium, bacteroidetes like Barnesiella, Rickenellacea, and then the microbial gene for indole production is that tryptophanase A, which they found if you knock out that gene, there was about a 5-fold reduction in CIA severity. So indole increases pro-inflammatory cytokines by altering the pathogenicity of autoantibodies. So without indole, antibodies had a reduction in activating complement, therefore lowering the amount of pro-inflammatory cytokines. And so this kind of suggests that antibody function is significantly more important in the severity of disease than the actual levels of antibody concentration. And so how does it alter the pathogenicity of these antibodies? Well, it does this by skewing T cells towards the Th17 line. This is mediated by IL-23, and Th17 cells release IL-21 and 22 that increase pathogenicity of antibodies. And so they found that this indole-mediated skewing of Th17 cells might actually be relevant to human intestinal immunity because they took human cells that were exposed to indole from intestinal samples, and they saw that the same increased expression of genes involved in the IL-17 signaling pathway was found in those cells as were in the mouse studies that they did. And so what can we do about this then? So can changes in your diet affect the probability of developing arthritis? Well, tryptophan is an essential amino acid, so it has to come in through your diet. So technically, yes, it should be found. It was found to benefit disease severity. This is due to lower tryptophan, meaning less tryptophan is available for microbial metabolism. You have less indole, and you then have decreased pathogenicity of antibodies. And so this quote comes from one of the main papers that I looked at when I was doing my research. We modulated indole production through either antibiotic-mediated microbiome depletion or reduction of dietary tryptophan. Depletion of either the microbiome or dietary tryptophan protected mice from developing CIA. So some examples of high and low tryptophan foods. High tryptophan foods are things like oats, milk and cheese, poultry like chicken and turkey, and then some examples of low tryptophan foods are things like apples and most fruits, almond milk. And then if you have like a plant-based diet, then you're typically lower in tryptophan as well. And so that is my presentation, and here are my references. Questions? Take the water. Yeah. Like there. Meaning, so if you have lower tryptophan, you're going to have a better outcome. You're not going to have as significant disease. So tryptophan is almost positive. Yeah, kind of. It potentially could. Often, from what I found, dysbiosis kind of acts more as like they haven't found whether or not dysbiosis is caused by the arthritis or arthritis causes the dysbiosis. So can't really give you much of a solid answer on that yet because they're still unsure which one causes which.